So just to recap, 3 types of RNA. mRNA is the first, and mRNA is what carries a copy of the genetic information from the DNA to the ribosomes. It's called mRNA because it carries that message of genetic instructions. rRNA is just a structural component of ribosomes. So RNA, rRNA, and proteins make up ribosomes. And tRNA is what you used to create your bead chains during that lab activity. You had a long mRNA strand, and you found the tRNA anticodon that matched it, and you figured out which amino acid that tRNA was bringing so that it could continue to bring more and more amino acids to create your polypeptide chain. All right. Next up, we're going to talk just a little bit more about transcription, and particularly the molecule Rna polymerase. Anytime you see the ending ase in a word, it means it's an enzyme. Anybody remember what a polymer is? So Rna polymerase builds RNA polymers. Another way to put that is, it's the molecule that creates RNA from the DNA template. I can see Evan over there wondering, how does RNA polymerase know where to start? I see it in your eyes, Evan. RNA polymerase knows where to start because there is a section of DNA called the promoter region. It's a very particular set of nucleotides all in a row. And when RNA polymerase finds that promoter region, RNA polymerase binds to it and starts copying, starts creating mRNA from the DNA template. Evan, how does RNA polymerase know when to stop? And it knows when to stop when it hits a section of code that doesn't code for a gene that's called a termination sequence. Once RNA polymerase hits that termination sequence, it stops making RNA. All right, last but certainly not least, we're going to talk a little more about translation. Then we will be done with notes. Well, we have to talk a little bit about mutations, but that's super quick. So, translation. When mRNA bonds to the ribosome, it is called initiation. So this is really just going to be vocabulary and definitions that you need to know in order to properly describe how translation occurs. So when mRNA bonds to the ribosome, it is called initiation. mRNA codes in 3 nucleotide segments known as codons. That's something you guys know. That's a term you've seen before. You use those codons to create your bead amino acid chains. tRNA codes in 3 nucleotide segments known as anticodons. You know that as well because you used those laminated tRNA that told you which amino acid was attached to figure out which amino acid to put in your chain. And each tRNA has a particular amino acid bonded to a particular anticodon. As the polypeptide chain grows, it's called elongation. Remember that a polypeptide chain is the technical term for a protein. And when a ribosome reaches a stop codon, that part of translation is called termination. It stops making a protein, it terminates that protein. All right, right now we're going to practice a skill you know using a tool you don't. So here is a generic mRNA codon sequence. I've broken it up into its codons so it's easier to see, easier to read. What I'd like you to do is figure out the tRNA anticodons that would bond to that. And just to remind you, A goes to U, U goes to A, G goes to C. That's what we're doing here. So you need to tell me what tRNA sequence would be bound to this mRNA sequence. Take 30 seconds and do that, and then I'll reveal the answer. All right, hopefully you got something like this. UAC, GUA, AGU, ACA, GAA. If you got that, this is a skill you've got. You'll see it on the practice quiz today. You'll see it on the real quiz tomorrow. You won't need to sweat that portion of it. Now what we're going to do is use the slightly different tool here. The circular tool we use to decode will be available to you tomorrow on the quiz. So the way this tool works is that because it says codons found in messenger RNA, we're not actually going to worry about our tRNA anticodon sequence. We needed that in case the tool we were using had tRNA for the sequence, but what we're using is the mRNA sequence. So the way we'll look at this is first, second, and third bases is how we'll use this tool. First base, second base is along the top, third base is along this side. So my first base is an A, so I look with the first base section over here. I find the A, first base could have been U or C or A or G. Because it's A, the amino acid I'm going to use will be found in this green box, that big A box. Then I look at the second base. So my second base is a U. The second bases are found up here. This is the U for second bases. So where the U meets inside my green box, that is the potential for which amino acid I'm going to be coding for. The last one is a G, so I'll look over here in my third bases. Here is the G inside of this green box where it can meet up, and where the G and the A and the U meet up is methionine. So I know that I'm coding for methionine. Okay, I'm going to do this one more time. If you feel like you got it, just go for it. If you would like to see it happen one more time, I'm going to do it one more time, and then I'm going to let you do the remaining 3 codons. Okay, so I'm going to erase all this. The next mRNA codon I'm looking at is CAU. So I'll look where the first base is. First base is a C. I circle my C. I know that it's going to be inside that green box, one of those 16 amino acids. My second base is an A, so I look up here in the second base area, and I find the A in the second base area. So I know that it's going to be inside that red box, where the C and the A meet up. And now I'm going to look at my third base. It's a U. I'm going to look in this little third base section here. I find the U right there. So where the U meets up is histine. So I know that my next amino acid is going to be histine. I'll leave that up there as you do the next 3 codons. Call me over if you have questions. Hopefully in using this new tool you got the next one was serine, then cysteine, then leucine. Remember on the quiz tomorrow you'll get to use whichever tool you prefer. On the practice quiz today I'm going to have you use this tool just to get a little more practice with it, because it seemed like everyone was doing well with that circular tool from the before Thanksgiving break.